Finally, the last speaker of the day will be Professor Gerd Müller, who is another world-famous theoretical biologist. And Gerd is also a colleague. He's an MD originally, but after his medical studies, he pursued studies in zoology, made his PhD in biology, and became an evolutionary biologist who contributed, again, with very pioneering and landmark publications. And he is regarded as one of the founding fathers of the Evo-Devo theory, and his famous book with Massimo Pigliucci, Evolution, the Extended Synthesis, is the latest grand work or the latest chapter in evolution theory. So the third speaker will be Gerd, who will explain to us one interesting consequence of everything we have been talking about during the past year. How evolution theory evolved and still evolves. So,
thank you very much, Balázs and Jan, for inviting me. I'm very honored to be speaking in this wonderful series that has been going on for so long. And I think you will have noticed that evolutionary biology is a field that has enormously diversified, and you get many different point of views. And as part of this epilogue, I will try to pull together some of the threads and especially return to the conceptual aspects of evolutionary theory and see how and in which way these new developments are actually influencing how we conceptualize evolution. And the reason for this is that in recent times there have been a number of debates and even quite heated debates going on that even made it into Nature about whether or not, in the light of new evidence, we have to rethink some of the aspects of what I would call the standard theory that still appears in the textbooks as the modern synthesis theory that you all will find in these textbooks. Now, it should not come as a surprise that this discussion is going on, because after all, this standard framework is more than 70 years old, and think about what had happened in the meantime. So I would argue it's not correct that some of the representatives of the standard framework say it's even fatuous to think about any incompleteness or any changes to the standards framework. For those of you who don't know the term fatuous, it means idiotic. Whereas others have pointed out that no theory is ever complete. Theories have always evolved. And if you look at the evolution of evolutionary theory, so to speak, it of course goes back to pre-scientific times. There were concepts that had been developed and incorporated in later versions of evolutionary theory. And, we're standing somewhere here with the modern synthesis. But the question is, why would the development of evolutionary theory stop in the 1930s or 1940s, whereas all other aspects of evolutionary biology have advanced, and just the theory itself would remain miraculously unaffected by this. So indeed, a number of significant challenges to evolutionary theory have been published in recent days, and I give you a little overview here, and there is many more, of course. And in this lecture tonight, I would like to condense some of these challenges and try to show that it is possible to actually have an expanded framework that takes into account these different perspectives. Before I do so, I would like to remind you that there are very different perspectives that you can take on evolution when you think about evolution. You could, or for example, think of evolution as species diversification, and the goal would be to reconstruct the tree of evolution, whether it's with comparative anatomy or whether you do it with genomics. But such fields as systematics and phylogenetics are mostly concerned with this kind of perspective. But you may also look at evolution as variation in populations, of course, and whether that's phenotypically or whether that's genetic, study how variation in population arises, how it is modified under certain selectional regimes, what kind of predictions we can make about this variation, and so on and so forth, which is fields that are covered by population genetics and quantitative genetics and so on. However, you may take even another aspect of evolution, which is the evolution of complexity, as we call it, of organismal complexity, where you see different types of body plans that are being established in the course of times that are very, very differently, architecturally differently constructed, or other phenotypes. You could replace this by behavior, for example, or by physiology, or by development, right? And there are the fields of evolutionary developmental biology, that's called Evo-Devo, behavioral biology, systems biology, and other research programs that address this kind of problem and these kinds of questions, right? And there is yet another perspective we might take, which is the evolution of language and mind and culture, which is mostly covered by the fields of anthropology, linguistics, social sciences, and so forth, and especially also the feedbacks of these kinds of properties into the evolutionary process. So we must be aware that the debate I was talking about takes place before this diversity of different approaches and actually different explananda. It depends on what we want to explain about evolution, in which way we look at it. And finally, as the end of my preamble, we should be aware that also the definitions of evolution have been changed over time. Starting with the most elegant definition, I would say, on the top by Darwin, descent with modification. This is fantastic, you know, but we have arrived more or less down here, where you find, even in the opening sentences of certain textbooks, that evolution is the change of gene frequencies in populations. And so we could say that this kind of development here has something to do really with the modern synthesis that was established in the 1930s and 1940s. And I would like to quickly, very, very quickly, much too quick for Ursh's, recapitulate the tenets, the central tenets of the modern synthesis theory, in order then to contrast it with other challenges that have arisen. And as you all know, the modern synthesis was a fusion, if you like, of concepts. It never really was a synthesis. It was more a semantic synthesis. But essentially neo-Darwinian theory, which incorporated already Mendelian genetics, but had eliminated the Lamarckian aspects of evolution, was fused with variation thinking in genetics, so covered by population genetics, experimental genetics, systematics came in, and then there were contributions also from the fields of paleontology and botany that had not been part of the previous evolutionary thinking, but they didn't contribute any new kinds of mechanisms or factors, but rather contributed to these other aspects that formed the modern synthesis in the 1930s and 1940s. But the core key principle, so to speak, of the modern synthesis is, of course, population genetics, right? And population genetics, you could say, is a mathematical account of the relations between genetic variation, inheritance, and reproductive success, mediated by natural selection, as it were. This is a very rough summary, but with the goal of explaining variation and adaptation of populations. So really, the modern synthesis is a theory about variation of components that you can measure and that you actually exist already in populations. So for example, you could never measure a novelty that does not yet exist because it's per definition is not there. And so population genetics and let's say this fitness landscape is a symbol, a representation of population genetics, really is the formalized core of evolutionary or of the modern synthesis theory. And it has a few central tenets that I would quickly read out to you. It's the only slide with a lot of writing on it. So one of the tenets is that populations contain genetic variation, of course, that arises randomly from mutations and recombination. And that populations evolve by changes in gene frequencies brought about, as it says here, by natural selection, gene flow, and drift. Okay? These genetic variants then, and that's very important, would have slight, minute, and gradual, continuous, incremental phenotypic effects that result in phenotypic variation and which by definition then is gradual. The graduality is a key component of this. You know, Darwin himself argued that if a single example could be given where a new trait has arisen by non-gradual evolution, my whole theory would fall down. Which of course is not true, but he felt it that way. And in the modern synthesis account, it's genetic inheritance alone that accounts for the transmission of selectable traits in subsequent generations. And it is natural selection that provides the only directional agent, so to speak, by acting on these small and incremental differences, and I mean, you could name many more, but I selected these central principles. And when you look at it this way, and you point out, you know, what the argument is, you see it's really an argument about genetic evolution. Genes figure prominently in all of these key components, and it's natural selection that acts on this genetic variation. So it's really, we could say, do these principles exist? Of course they exist, yes? Are they sufficient for a full account of the phenomena we observe in evolution? Well, no, because we could say the modern synthesis theory really does very well what it has been constructed for, namely to address, you know, variation in populations, address or predict gene frequency changes in populations under certain regimes. It explains gradual variation of phenotypic traits and the adaptation of such traits, and it explains certain important genetic features of speciation. However, there is a number of phenomena that we need also to address, such as the phenomenon of complexity. And so the standard theory does not contain a theory about phenotypic evolution, about phenotypic complexity, about the fact that not all variants that can arise in the population are equally likely to arise. There is the eternal question of how phenotypic novelties arise, how non-gradual forms of transitions can occur that are documented in the fossil record, or how non-genetic factors of change figure in that theoretical framework. So we could say that the standard synthesis that we find in the textbooks that's still dominant is actually a theory about variation and adaptation in populations. Therefore, I believe this is the very reason why increasingly challenges from other fields that fully accept the population theoretical account are being mounted and proposed. And so let me tell you about a few of these challenges. And I start out with the field of evolutionary developmental biology, in which I'm myself active, and Sydney actually has been one of, you could say, one of the founders, even if he might not know that it's evolutionary developmental biology. And I would like to begin with explaining that Evo-Devo, evolutionary developmental biology, is really a consists of a dialectical set of questions. On the one hand, you're asking, how is evolution affecting developmental processes in order to provide change, because organisms do not change because the genes change, but because their developmental programs that are establishing the structures are changing. And this, you could say, is actually mostly about the evolution of development, how something evolves, right? Especially it's the cookie problem that Sydney has mentioned, and I would say 95% of all of evolutionary developmental biology are doing this by comparing gene expression patterns in different organisms and looking at, such as in this case, at Hox gene expressions. And one of the most wonderful discoveries was that the Hox genes are largely conserved, duplicated and everything, but they're involved in the regulation of completely different sets of organs in the different organismal lineages, which is very important. Or another way of looking at the evolution of development is to try to understand how the regulation of developmental processes is evolving itself, which interactions are being modified, and so on and so forth. So that's very well, and that's pursued at a large scale, because Evo-Devo has become a very institutionalized field with institutes, professorships, grant applications, journals, books, conferences, societies, and so on. So that is a big enterprise. You shouldn't think that Evo-Devo is something minor. It's very actively pursued. But what we're interested in today is rather the lower aspect of these sets of questions, which is asking, how do the properties of developmental systems actually influence the evolutionary process, influence what can happen in evolution. And this is, if you like, the role of development in evolution, and this is where the concepts that pertain to evolutionary theory are actually become important. And the Evo-Devo field has been very prolific, you could say, in generating such concepts. And here is a little selection of it. And there are very interesting things. All of these have empirical evidence. It's not something that's been invented. For all of these, multiple publications exist. But I'm going to select 3 principles only, which I believe are the 3 most important ones. And the first one is developmental bias. So the variation that is possible to be produced by an organism and its population is actually not equally likely in all dimensions of variation. Here you have a wonderful example regarding the eyes of these butterfly wings, where you can see that, as Brakefield has demonstrated, that selection in this axis, towards making the eyes bigger or smaller, in which case one of the eyes disappears, is very easy to achieve by selection experiments. But variation in this axis is absolutely impossible to achieve, where either the posterior eye is made larger and the anterior one should disappear, or vice versa, you would want to make the anterior one larger and the posterior one smaller. This is not possible. The variational plasticity of the system cannot produce it. Yes, he called this wonderfully the anisotropic accessibility in morphospace. This is I should remember this. Another important principle is that not all of developmental variation is deterministic. Rather, by various kinds of processes, of which one of them is shown here, which is the Turing mechanism, you can see that if you have 2... Okay. So, Turing, whom you may know for quite different things, he wrote one paper in biology about morphogenesis and morphogenetic dynamics, which has become a classic and is incredibly important. And he showed that if you have 2 substances, morphogens as we might call them, diffusing against each other, you create certain patterns of interference of higher and lower concentrations of those morphogens prefigure a pattern upon which the cells organize themselves. And you get these wonderful surface patterns through these mechanisms, very nicely researched. And this is a principle in which the individual position of individual cells is not determined. Rather, they follow the stochastic arrangement principles, and so not everything in development is a strict consequence of a genetic program, of course. And thirdly, what is also important in Evo-Devo, or an important outcome of Evo-Devo research, is that not all developmental variation is continuous. So the variation, the selectable variation that can be produced by these organisms depends on highly discontinuous mechanisms in development, such as this one in the formation of somites, where the individual cells can either fall into an FGF-rich domain or into an retinoic acid-rich domain. And you may realize Goldbater's work here. Which means that development and its variation in evolution is affected by what we call bistabilities of these systems. And therefore, if you have natural selection acting on such a system that contains lots of bistabilities, the outcome by necessity cannot always be gradual and continuous and incremental. So this may have been very theoretical, and therefore I would like to give you an example. And the example is from our work on limb development. And you can see here that limbs in development and in evolution are characterized by either the loss or the gain of digits, if we assume that the pentadactyl pattern is the basic limb pattern that has been established. This can be questionable, but it doesn't change the question or the problem. So limb development is based on a very well-known deciphered interaction for molecular regulation. This is a limb bud, these are the cells, and the cell proliferation is regulated by an interaction of, certain factors, Wnt and FGFs and sonic hedgehog and so on. But this is the same regulatory interaction that is true in all these different limbs. This is true for all tetrapod limbs, right? And it acts upon another mechanism which you could call cell autonomous, which means that when cells aggregate together and they reach a certain threshold number of cells that come together, they switch over to cartilage matrix production. You can do this in vivo, you can do it in vitro. You have more than 400, 450 cells, and then they form these cartilage nodules. So the formation of these individual skeletal elements depends on the sufficient number of cells coming together in order to initiate that cartilage differentiation pathway, and matrix production goes up immediately. So it's very interesting to understand. Now the pictures have disappeared. We are back. Yes, okay, let's stop a little. Okay, yes, thank you, wonderful. That's much easier. Now you can modify this system by either evolutionary or experimental perturbation. Let's look at evolution. There is a wonderful example here about digit variation in Australian skinks. And the fantastic thing is they have hundreds of species. And in many of these species, you get limb reduction and certain in-between stages of reduced limbs. So you could say there is natural selection acting against limb size. And they assume a more snake-like behavior. But the outcome is that these are different species. It's a morphocline. This is not one selectional effect, but it repeatedly, if you get digit loss together with the reduction of limb size, it will always be digit number 1 that disappears first. Second, digit number 5 will disappear. Third, digit number 2 is going to disappear, and so on until only a remnant of digit 4. So this will always happen, okay? And note a second aspect of this. It's full digits that disappear at once. You would intuitively assume if you have selection for limb size reduction, you start with a reduction of the digits of the phalanges from the distal tip. But this is not the case. The first thing that happens is a full digit disappears, right? So this is a very nice example for a biased response of the system. It's always anterior. You can predict the sequence of digit reduction if you know how the system works, because in development of reptiles, the generation, the appearance of digit is the exact reverse of the disappearance pattern. So you can, if you know this, you can say if limbs are reduced, and I go out into the Australian bush and I collect skinks, I can predict what I'm going to find. So this is something, for example, that population genetics couldn't do in this case. But you can also experimentally test this by making the animals smaller. And how do you make animals smaller? You can arrest a few cell cycles by a mitotic inhibitor, and we have done this, long ago in Mississippi. You can make the alligator smaller by small doses. If you raise the dose, the alligator gets smaller and smaller and smaller. But the outcome is, if you look at the limbs, that the digits start to disappear. And here, precisely as predicted, with a small dose of a cytosine arabinoside, a full digit has gone, and it's number 1, right? And if you increase the dose and body weight goes down, you have certain threshold levels, and the next one to disappear is number 5, I'm sorry, and then 2, and then you get a lot of pathology and it doesn't work anymore. But you can reproduce experimentally the same bistability, so to speak, where the variational system of limb bud formation reacts in one specific way. And you can also do the opposite. You can add digits experimentally. For example, by natural mutants or by these transgenic mice, where you can see these are additional digits. And again, it's full digits that appear, not a little rudiment that may later be elaborated or so on. But the system produces immediately full digits in various kinds of transgenic mice. And we studied an interesting case that's well known, which is the Maine Coon cat, a species of cat that's raised and lives in Maine, in the province of Maine. And they have frequently preaxial polydactyly, as you can see here. And this is based on a single point mutation in a conserved non-coding element that is part of a cis regulatory region for a protein called sonic hedgehog. And sonic hedgehog is very important in setting up the polarity of limb development. And it's this mutation that the cats have here. So there's A to G, which I believe Sydney said doesn't happen. But it's called the Hemingway mutation because the cats in Hemingway's house at Key West, they also frequently have this polydactyly. So what happens is that this mutation interferes with sonic hedgehog expression, and you can see in the mutants here that sonic hedgehog is expressed in anterior regions of the limb bud where it's not expressed in the control limbs. And the outcome of this, you can now look up in wonderful databases that the breeders of such cats keep, right? And you can see exactly how many digits they have and so on. And we actually looked at 485 Hemingway mutants with preaxial polydactyly. And what you see is that the results are polyphenic. So you can get 1 digit, you can get 2 digits, you can get up to 8 additional digits in a single cat with the same point mutation. And they don't appear with the same frequencies. The normal wild-type number of digits is 18, 5 on the forelimbs, 4 on the hind limbs. And you see, except with this a little elongation of the thumb. But all others, happen in the same pattern as I've described for the mutants. And you see that the most frequent is the 6, 6, 4, 4. So nothing happens in the hind limbs, but there are 2 additional digits in the forelimbs. So this is not a normal distribution, right? This is a biased distribution of these effects. So the effects are not by chance. And we were very interested in how these thresholds actually appear. In why is it? It's not only anterior, it's always anterior, as you can see, but it also happens in a stepwise fashion. And we looked at that and tried to construct a model for digit formation and threshold that appears in digit formation. And we did this when I cut it very short because we don't have enough time. But if you take the cells in the limb bud as a binary switch, each cell that can either enter the cartilage differentiation pathway or the non-cartilage differentiation pathway, then you can construct a cellular automaton in which cells are in a matrix, you know, and they can either be activated or non-activated. And you have a certain seed value, and it all depends on a reaction diffusion Turing mechanism, so to speak, between activated and non-activated cells. And you let the system run, and then you see that there are certain regions of activation, which are the bright regions, and regions that are non-activated, and you let it run long enough, and you get wonderful striped activation stripes that we could, so to speak, liken to digit formation if we assume that the cells in the limb bud are also act as binary switches. And now you can vary certain parameters in this system. And I don't go through the whole story, but if you, the best predictor for polydactyly is actually the variation of reaction rate, which is the relation between activation and non-activation. And you see that the stripe formation follows, you know, a stepwise pattern, but the threshold widths that you have here are different. And they are different in every run. These are several runs. And you see that sometimes the threshold is very narrow, like here or here. So they actually go from 6 digits to 8 in one step, practically one step, right? So this all happens. And now if you do an exponentiation of this reaction rate, you can actually have a prediction of the thresholds that exist in such a system that exists of binary switches. And if we then superimpose our results from the cats, then you see that they fit quite nicely along those predicted threshold patterns. So what I mean to say is that such systems that are biased and highly nonlinear must influence the variational behavior of traits in populations. And so you could say that developmental systems can react to selectional or environmental perturbations in biased, emergent, and discontinuous ways, and such introducing nonlinear variations into the evolutionary process. And the second most important consequence is that we can predict, you know, knowing the developmental system, the behavior of them, we can predict which kinds of variations can be produced by such a system. So this is quite different. And now let me add a few other challenges from other fields in a sweeping survey of field. But there's lots going on. And something Sydney has talked about is, of course, that genomic evolution behaves quite differently from what has been assumed like 50 years ago. So it's not single allelic substitutions that accumulate slowly that are really important for phenotypic evolution. But we know that gene duplication is pervasive. We know that horizontal transfer exists. We know that epigenetic modification exists, and so on and so forth. So the dynamics of genetic change are quite different from what has been assumed in the standard picture. Another aspect is the levels of selection issue. In the standard view, it's the individual that is the level of selection. But in the meantime, we know that at least kin selection and group selection are very likely. The verdict on species selection is out. But we also have infra-individual forms of selection. And a certain speaker who went before me actually presented a model that spans several levels of selection. And I do not now have to explain the replicator model here once more, but this is such an example. We have inheritance theory, and in the standard view, it is genetic inheritance alone that accounts for the transmission of selectable traits. We know that there is epigenetic inheritance, whatever its meaning might be in evolutionary change, but it exists. This is a fact, you know, transgenerational epigenetic. We know there is inheritance through learning, and we know there is cultural inheritance, and altogether this has been called inclusive inheritance, which is, if you like, a step forward from a single genetic inheritance. Finally, there is something that is very interesting, namely that environmental effects can induce directly developmental modifications through direct influences on the variation of and the plasticity of the developing embryo, and by, so to speak, by bypassing natural selection. But natural selection would act on the outcomes of this type of variation, but that is induced environmentally. An interesting concept being mostly proposed by Mary Jane West Eberhard in her famous book. Then there is an increasingly important concept of niche construction, which more or less explains that populations of organisms are not passive recipients of selectional processes, but populations of organisms actively modify the environment that becomes the selective environment for the subsequent generations. So they generate the selective environment through their activity. And this even has effects on genetic composition of populations and so on. It's a concept that's been called niche construction and has become very important now. And finally, I would call in systems biology, not the omics type of systems biology, but the sort of multi-level organizations systems biology, mostly covered by physiology and functional biology. That spans different levels of organization and challenges very much the gene deterministic program without going further into it, but there are wonderful papers on that. So we can say, as a consequence, that some of the essential tenets of the modern synthesis have stopped to serve as the unique explanations. They are not wrong, but random variation, individual substitution concepts, single level selection, uniquely genetic inheritance, and so are clearly superseded by a more pluralistic attitude. So now what can we do about this? Is there a way of theoretical integration of these developments? And I'll show you a possibility. Well, let's first look at the concepts. You know, we could say that, you know, the modern synthesis exists of a collection of concepts that include the Darwinian concepts. And we could simply add now, you know, those concepts that I've mentioned, but also others, of course, and say, yes, these new concepts, they may exist, but they are a peripheral addition, you know, to special cases, and they are not so important. So my question here would be, does this, you know, incorporation of such theoretical concepts influence the structure and the logic of the evolutionary argument in any ways? And I'll try to show you with in a cartoonish way, because I cannot do it now differently in such a quick time, that I believe they do. So if we start out with a depiction of the classical modern synthesis view, where we say there is populations of varying phenotypes and their traits are influenced by the gene pool of the population, right? The variation is influenced by the gene pool, and the argument would be that natural selection is acting on the outside, from the outside externally on these populations, and inheritance between generations would be uniquely genetic. I would argue that first, this aspect of the gene pool here, which is marked in yellow, should be replaced by what we might call a developmental pool. So it's development with all its forms and incorporation of non-genetic, like self-organizational aspects and so on and so forth, that actually determines what kind of variation can arise in gene pools, in phenotypes, in populations of phenotypes. Now, then natural selection we might be able to replace by multi-level selection, although formally this doesn't have so much. But this is important, that if we add niche construction to this, we have a kind of reciprocity that arises here, and not just a unilinear causation of variation in populations. We can also add the different forms of inheritance here, and we have a multitude of possibilities for inheritance between generations, and we can add environmental induction that acts directly on different forms of phenotypes. Now, I know these are arrows in a diagram, you know, but it should just illustrate to you that we have, if we do this, and the other aspects could be added, if we do this, we have a much more complex picture than the one we started out with. And it has a number of very important differences that are, you know, you find here, 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 and here in these four aspects. So we could make an argument that through the incorporation of such concepts that arise from very different fields, the internal logic of the evolutionary argument is in fact affected and is changed. And we might say, okay, so what? You know, does it make any difference? And I would say, yes, it makes a difference because the predictions that result from this kind of approach are different from the standard predictions. And I've made here a little comparison, a table also, very, you know, an overview. But you could say that variation in the extended view is biased and determined by development. the selectional process may be always going on, but the specificity of the developmental response, is what defines the phenotypic outcome. Genetic evolution in the standard view is actually kind of driving phenotypic evolution along, whereas in the extended view, it rather is used to stabilize emergent components that will happen automatically if you modify developmental systems. Inheritance, as we said, would be inclusive. The rates of change would, of course, often be gradual and continuous, but the extended view includes the possibility of having discontinuous forms of variation, as we can see in the fossil record very often. Natural selection is a long story. It would need its own lecture, but in the standard view is mostly interpreting the maintenance of phenotypic variants, whereas in the extended view, the natural selection could be interpreted as something that releases developmental potential and then can be stabilized and go to fixation. The environment here would be independent of organismal activity, whereas in the extended view, the environment is generated by organismal activity. So this is simplistic, but it needs to be fast when we go here. So I would say that this kind of view, and by the way, no one is calling for a new theory. What I'm saying is that the concepts we are working with and that the different laboratories in the world are actually using are different from what the standard theory has been prescribing, so to speak. So whether we call it extended synthesis or whatever, it really doesn't matter. I'm saying we're undergoing a period of conceptual change regarding the composition of the theoretical framework. So we have, an expanded explanatory reach regarding the genotype-phenotype relation, origin of complex traits. I would say some of the concepts underlying major transitions are affected by this view. We can explain more punctuated events, behavior, language, and gene culture co-evolution. So I would say that what has been termed the extended synthesis, but could be called anything else, is not an idea of a few people who would like to revolutionize evolutionary theory, but it comes from new data and new concepts that have arisen independently in different fields, and it could be possible to bring them together. And the ES integrates some of these concepts into a coherent theoretical framework, and most importantly, the predictions of this framework are different from the predictions that result from the standard framework. And we could say that therefore, this concept has important heuristic values by making new predictions that can be tested experimentally, and therefore is of importance. Now, if I have 2 more minutes, I would like to show you something which I have added especially for Balázs, some of the future directions that this might go. Now, one of the necessary future requirements is formalization, of course. So I do not believe that there will be a formalized theory that can incorporate all of these components. This is quite unlikely. But certain aspects can be formalized, and it has already been done. Like in the case of inclusive inheritance, there are very nice proposals how to distinguish between genetic and non-genetic parts of inheritance and to divide the non-genetic up in yet different components of inheritance. Very nice work by Etienne Danchin proposing this kind of. But I see, for me, for my personal, view, a different kind of formalization would be required, and that is to bring the population theory in relation to what I would call the organization theory, which is essentially, the relation between genes, cells, and development that create complexity. And in order to do that, what you need is quantification of developmental processes. I would say developmental processes lag behind in the. There's a lot of description. There's a lot of discrete results, cookies, and in our laboratory, what we're attempting is to develop quantitative methods to measure developmental processes and compare them. And the way we're doing this is actually by developing micro-CT techniques for embryonic development. Now, you have shown us your wonderful scanners. Here is one. But our samples are here. This is the sample. And the embryo that we're scanning is maybe 2 millimeters in size, right? We're doing this with micro-CT, the problem being that embryos consist 95% of water, and you have to do something to enhance the contrast, but we have been very successful in doing this, and you get to a resolution of almost cellular level, and this is already, this is a few years old. We have much better resolutions in the meantime, and we even went on to do micro-CT with molecular probes, where we can, stain. This, for example, has been stained for tubulin, and this is maybe 2 millimeters in size. So we have, methods to quantify. The beauty of this is it looks beautiful, but the real beauty is it's all numbers. And you can calculate anything you want to do. And the idea being that you do large numbers of embryos and measure the variational components. And then actually you could apply similar algorithms, such as in population biology, to the variational properties of embryos. And we have done this, you can quantify gene expression, for example, which is really important because gene expression is not only a topological problem. It's a problem of how much, at which time, where, and so on. And this defines the variational properties that become important in evolution. So I'm finishing by saying this is not a one-man show that has somehow invented extended synthesis. There are very largely funded projects underway. One by the Templeton Foundation that is 8 million worth. And there is lots of labs worldwide that are part of this effort and this approach. And with that, I would like to thank you for your attention and for being here. Thank you very much.